Okay, hey guys. Step one when you're making a video podcast for your students is to press record. Here I go. Let me roll through this presentation for the second time, but this time I'm going to record it. Uh, Molecular genetics. I want to roll through this with you guys. I explained it in class of why I want to try this format. First and foremost, it's to save us some time so we can do some interesting labs in class, and we have enough time to do that. I think I think it's best to use our class time to do science instead of just talking about it. So that's the goal of this. I'm interested in your feedback to see if this is working for you. It's something I'm happy to continue certainly doing if it's working for you guys and it's helping us learn better. But I need that feedback, so just let me know. But let's give this a go. So molecular genetics. I'm going to attempt to do a few things. Come on. There it goes. In this video, in this podcast here. Make sure that you guys have those slides and that you're annotating those slides as I'm going through. This is going to be useful for you if you have those and you're annotating so sort of currently and actively. I'm going to go through Darwin's ideas of natural selection with molecular genetics. How do they work together? So we just finished talking about evolution. How's molecular genetics going to tie into what we're talking about in order to create something called the modern synthesis, which is the domain in which we are working right now? The discovery of DNA and who contributed to that discovery. And also the structure and the function of DNA. How does how DNA is built lead to its job that it does for us? Okay, so feel free to pause the video, stop it when you need to. I'm going to go quickly through these. I have 15 minutes to present to you guys before they cut me off here with the free version of Screencast-O-Matic. So let's go. Darwin's ideas of natural selection. How they work together with genetics is what we call the modern synthesis. Darwin had no idea what the inheritance molecule was. He didn't have the vernacular of gene or allele or DNA when he was doing his work. He knew there must have been some sort of inheritance molecule, but had no idea what this thing looked like. So it wasn't that his ideas were wrong. It's that his ideas were lacking some details that we hadn't discovered yet. Those new details, along with Darwin's ideas, are what we call the modern synthesis. Okay, so this is really what we're trying to do and what we're focusing on. We're looking at where traits come from, and that's really from DNA and heredity, through some of the work of Gregor Mendel, which we talk about in heredity, our last unit in the modern synthesis. Where we're focusing now is how does DNA get turned into a usable copy we call RNA that then gets translated into some physical trait, usually represented as a protein. If you have that, then you have the raw ingredients necessary when you add variation and a little bit of competition in order to have Darwin's idea, which is selection. If you have all those ingredients working together, you have this new idea of evolution. And we have some examples of this new idea of evolution that we saw as we were looking at Hardy-Weinberg equilibrium and we were talking about alleles. Allele was not a word that Darwin had to throw around. Okay, so two major shortcomings in Darwin's theory of evolution: how variation arises. He knew it did, but how did it arise? That's through DNA. And then how inheritance is passed along, and that's also through DNA. This idea of neo-Darwinian evolution is the modern synthesis. Okay, our main focus of why we're talking about DNA, it is the inheritance molecule and the information dense packed molecule. There's plenty of ways that we can think about information in all sorts of different contexts. One of those contexts might be through books. What we do as a species, and something that we've been very successful at, is building upon our knowledge from generation to generation by passing along information from generation to generation in the form of language and in the form of writing. Books is a great example of how we pass information, specific information, down from generation to generation that tells a story. Computers also do this through binary code, a series of zeros and ones that look like nonsense, but when you put it together, allows you to watch the video that you're watching right now. And finally, the way that biological systems use information is through the molecule of DNA. So we can look at how information is used in all sorts of different ways. If I had to narrow it down and say what information really does for us, it allows progress. It is the engine of progress. The passing along of information leads to all the complexity, beauty, and unity that we see in our world today. And it leads to this cumulative effect and understanding of using our past experiences and passing that along from generation to generation. That's how we get to where we are today. DNA is the information of biology, and that's what we're going to focus on. Okay, so let's talk about its discovery. I'll run through this quickly. Feel free to ask me any questions about this stuff in class. The first person that we want to focus on is Frederick Griffith. Your book goes into quite a bit of detail with Griffith. He had this famous experiment with mice and streptococcus, a type of bacteria. The words I want to point out here are virulent and non-virulent. Virulent means it's going to get you sick and you're going to die. Non-virulent means it's not a threat to the immune system. So we had these two strains, the rough strain, which is non-virulent, and he said, I'm going to inject the mouse. No surprise, the mouse should live because it's not a virulent strain. Okay, so that's not a surprise when he injected this mouse, the mouse was alive. And then also not a surprise that when you inject something with something nasty that it could die. So these first two are not surprising, that you have a rough strain that doesn't affect the mouse and a smooth strain, which is virulent, and it kills the mouse. Then what he did is he took that nasty smooth strain and he heat killed it. He added a lot of heat until the bacteria was basically destroyed. And when he did that, the mouse lived. Awesome. What's really interesting, though, and the most interesting result of his experiment is what happened when he mixed the two things that had no impact on killing the mouse, the rough strain and also the heat-killed smooth strain. And what he found is the mouse died. His question was, why? Why did I see that the mouse died here? There must be something being passed along from this heat-killed smooth strain to the rough strain, and he was right. DNA was being exchanged from this bacteria to this bacteria here in a process called transformation, where bacteria exchange plasmids, certain bits of DNA with one another. He didn't know that, and he couldn't use those words, but what he did know is that there was some molecule being passed along from these guys to these guys, right? So that was sort of first steps into saying there must be some molecule that's being passed along in terms of inheritance. Avery, McCarty, MacLeod, I'll talk about them very briefly. They did some experiments in the 1940s that built on and added to the suggestion that DNA was the hereditary molecule. They had some confirmation using some of the same methods found back here with Frederick Griffith. One thing that I want to notice or point out, and why we should mention these guys, is that science, you don't get to just have one experiment sort of and make a conclusion and say, well, this is a brand new theory now and this is the way we're going to roll forward with science. It takes a bunch of different experiments that are going to confirm a theory over and over and over again, or a hypothesis over and over and over again in order to form a theory, which this theory we're talking about, DNA being the hereditary molecule. Hershey and Chase was sort of the last stop, I suppose you could look at, and when people started to accept, okay, it's nucleic acid that's responsible for information and inheritance. What they did is they took these little guys that look like little alien ships called bacteriophages. They're viruses that infect bacteria specifically. They're made out of two very simple things. Number one, they're made out of protein on the outside that make up their bodies, and they have nucleic acids on the inside. What Hershey and Chase did is they radioactively labeled the protein portion of the virus, and they radioactively labeled the nucleic acid portion of the virus. They injected and they let the virus do their things on bacteria, and then later, after they did all sorts of stuff like centrifuging these bacteria, what they found was the bacteria were not infected with radioactively labeled sulfur that was on the protein, but what they were infected with was the nucleic acid that was on the inside of those bacteriophages. So this led to the conclusion that it must be nucleic acid. That's the inheritance molecule. That was the work of Hershey and Chase. Chargaff is actually something you guys are familiar with. His famous work is on the base pairing rules, that A always pairs with T and G always pairs with C. The way he determined that is he ran a series of experiments and he looked at the frequency of how often adenine popped up, how often guanine and cytosine and thymine popped up in the genetic makeup of these different organisms. And what he found was that there always was a constant frequency between how often adenine showed up and thymine showed up, and how often guanine showed up and how often cytosine showed up. Meaning that for every adenine that you have in the genome here, you also have a thymine in the genome. And you could deduce that A must match up with T then. A is always around when T is always around, and G is always around when C is around. And he was right that these are how these particular nitrogenous bases pair up with one another. This is just another image of how they match and pair up. These two guys are generally who are given the credit for the discovery of the structure of DNA. I guess I've got nothing against these guys, but one person I do want to point out and I feel like is not given credit nearly as much as what she should is Rosalind Franklin. So a quick aside here about Rosalind Franklin. She was critical to the understanding and the finding of the double helix of DNA in the structure. Watson and Crick took her evidence and her data. They applied it for themselves because she was a good scientist and shared her data with the scientific community, but they failed to give her credit. She had already passed away from cancer by the time they had awarded a Nobel Prize, and you can't award a Nobel Prize posthumously. So she missed out on the Nobel Prize, which is a shame because she took this beautiful picture here using X-ray crystallography, which she was so great at. She worked in Wilkins' lab in order to do this, in order to determine the structure of DNA. Watson and Crick used that information in order to determine their structure and come up with their model. So yeah, they did a lot of the work, but so did Rosalind Franklin, and she deserves some credit. There's a great video here by SciShow. Hank Green goes through it, in which he talks about Rosalind Franklin and what an important person she was, and I put a lot of emphasis on her, and I think she's very worthy of conversation and our attention. So please watch that video also. Okay, the structure and function of DNA. This is the meat and potato sort of the podcast here. I want to walk through a few parts with you guys. I want to use an analogy here. If you've seen paper clips linked together in a long chain, that long chain is something I could refer to as a polymer. It's made up of many repeating units. Those individual units are individual paper clips. DNA does the same thing. It is a polymer of a bunch of monomers linked together that we call nucleotides. Those nucleotides then have three parts. The three parts are the nitrogenous base, which you can see here in this ring structure. The sugar, this pentose sugar, it's called a pentose sugar because it has 1, 2, 3, 4, 5 carbons. And then also this phosphate group that's sitting over here. This is one individual paperclip. This is the paperclip chain. It is a bunch of these nucleotides linked together. Okay. What's actually interesting is you guys have heard of nucleotides before, and you've actually heard of these nucleoside triphosphates before too. A great example is ATP. It's how it exists in your cells. So before it's added and incorporated and used in DNA, it's actually in the form of something like ADP or ATP, those really energy dense molecules that you guys learned probably in general bio. They're packed with energy because of these phosphate groups not wanting to be around one another. If you had one of these nitrogenous bases and you have one of these phosphate groups, you got yourself AMP, or you've got something that can be used in DNA. These are the different structures of the bases: adenine and thymine, guanine and cytosine. I'm rushing really fast now, guys, because I've got a limit of 15 minutes and with the free version. So I'm going to make sure that I get through these slides. What do we need to know from this slide? Well, that the purines are these double ring structures of adenine and guanine. Notice there's 1, 2, 1, 2 rings. Thymine, 1, 1 ring. So we give them different names to categorize them, the purines and the pyrimidines. Some differences between DNA and RNA that we need to take a look at. This is the pentose sugar that makes up a nucleotide. Notice attached to this 2' carbon, we've got a hydroxyl group. We've got a hydrogen group. Minus the oxygen, that's where it gets its name deoxyribose from. DNA versus RNA and some of the differences between these, other than that sugar that I just showed you here, and that there's a oxygen missing in the 2' spot for deoxyribose. DNA uses the nitrogenous bases A, T, C, and G, where RNA uses uracil instead of thymine. As you can see right here, this is thymine for DNA, and here's uracil for the RNA. And then the other obvious part of this is DNA is double-stranded, where RNA is single-stranded, and that comes in handy and leads to some versatility that RNA has that DNA lacks. A couple of the bonds here that are important to mention. There's 2 bonds that we need to know that involve just nucleic acids: hydrogen bonding and phosphodiester bonding. Phosphodiester bonds are the covalent bonds that join up a phosphate group and a sugar that allow the backbone to link together. So this is a better picture of that. You can see that there's this group, this sugar group, that links up with this phosphate group. These donate 2 hydrogens and an oxygen. They then covalently share this oxygen, and that's called a phosphodiester bond, and how the backbone links together. Individual bases are linked together with hydrogen bonds. There's 2 between adenine and thymine, and there's 3 between guanine and cytosine. This is just some of the notes about nucleic acid organization. I'm very close to running out of time, so I want to wrap up this video. You guys can read and look over those. Pause the video if you need to. These are the things you want to make sure you can do. Okay, guys? So it was a quick video, quick 15 minutes. Come loaded with questions, and make sure that you have your notes annotated and ready to go. Hopefully we have a quick 10, 15-minute discussion, and then we cut you off in the lab and do something fun. Okay, guys, thanks for watching, and give me some feedback. Let me know how this was.